Can you see my screen, doctors?
Yes, sir.
Yes.
Okay, so let's start. So the first is we are going to start from the topic. The first topic is complement system. So complement system is actually a system of hepatically synthesized plasma protein that play a role in innate immunity and inflammation. And inflammation. So this is the definitions of the complement system. So the complements are actually complement are actually proteins made in liver, and they will circulate. The liver will then excrete it out into the blood, and they will circulate in inactive form and get. So remember that when get activated, it will produce in a cascade. Means that one complement is then activated, it will activate the another one, and as a result a cascade of the complements activation take place, and this will play a part in innate immunity. So the final product will be MAC, which is membrane associated membrane attack complex, and it is associated with killing of bacteria, especially gram negative one, most commonly, especially. Okay, doctors. So this is the general information regarding complement system. So let's discuss the pathways that play a that are the part of the complement system. So the first pathway is classical pathway. So the classical pathway is, remember that its activation is usually occurred by two types of antibodies, that is IgG and IgM. So the IgM is actually a potent one. So this is the two antibodies that will activate the classical pathway, and its trigger is that formation of antigen antibody complex. So actually these antibodies will combine with different types of antigen, and then as a result of this antigen antibody interaction, it will start the complement system via classical pathway. So the antibodies that are usually involved in this are these two, IgG and IgM, and the IgM is the most potent. Okay, doctors. So you should also remember it that with the mnemonic, the classical pathway is activated like General Motors make classic cars. So remember that G for IgG, M for IgM, and classical pathway. So IgG, IgM will activate the classical pathway. So the steps that are usually involved in this are when antigen antibody complexes are usually formed, it will then activate C1. This C1 will then activate C4 and C2, and as a result the end product will be C4B2A. So this is the product, and this the another name for this C4B2A is C3 convertase. So actually we have to activate this C3 convertase, which will then follow the whole path. So this is called classical pathway. So let's move to the next pathway, which is called alternative.
Doctor, can you please repeat the first one, the classical?
The classic one.
Yes.
So the classic one, the main triggers for a classic one is usually two types of antibodies. It will only activated by antibodies. The two antibodies are IgG and IgM. The IgM is a more potent, and when these antibodies is combined with antigen, they are usually presented to the complement system and they will activate the complement system like in this manner. IgG and antibodies will combine, they will activate C1, and then C1 will activate C4 and C2, and this C4B and 2A will form a complex. This complex is called C3 convertase. And we're going to discuss that how this C3 convertase will work,
Okay.
because up to this all these three pathways will eventually converted into C3 convertase, and then after conversion into C3 convertase, the rest of the function of all these pathways are usually similar. Okay, that's why we're gonna discuss all the three pathways functions in a same way in the last. So the second one is alternative pathway. So the alternative pathway is activated via microbe surface molecule. So it is directly activated by bacterial surface, and here no antibody required. Okay, doctors. So it is work again. So this pathway will work well against gram negative bacteria, because these gram negative bacteria will have a specific type of lipopolysaccharides, and these lipopolysaccharides will activate this alternative pathway. The third one is lectin pathway. So the lectin pathway is actually activated by mannose, which are sugars or other sugars that are present on microbe surface. And the triggers for this are mannose binding lectins, lectin, it is a first, and recognize sugar on microbe surface, and it will have a similar pathway as that of classic, but it does not require no antibody required here, and use mannose instead of C1 as in classical pathway. Okay, doctors. So this is the three pathways that are usually come with us, and these three pathways will usually eventually form C3 convertase with us. So let's draw a diagram regarding these three pathways. So classic and lectin and alternate. So classic will activated via C4b2A receptor, and this is called C3 convertase. And this alternative will be also C3b BB. This will also called C3 convertase. Actually, from there the whole story starts, that after that we have two different types of like C3b activations, and this is usually responsible for opsonization and MEC activations that will usually required for membrane activity. So see, this C3b will then combine with this C4b2A 3b. This will again activated are in this manner, like C3b BB C3 BB3B. So these both will then have activate. So this is called C5 convertase, and this C5 convertase is then activated into C5b, and this C5b then combine with four other complements protein, that is C5 to C9, and this will then activate MAC, and this MAC is responsible for making pores in the bacteria and lead to lysis. So we have discussed earlier that C3b is required for. So actually, oh,
Do
yes,
we
doctor.
have to do we have to just memorize all this like C4B2A,
No,
like
no,
just
no.
or just no
I
need?
will tell you. I will now. I
Okay.
will tell you the important one. Okay. Okay, doctor. So in this, only the important one is this C3b, because this C3b is actually a role in opsonization, and opsonization means that this C3b will coat bacteria, and it will lead to enhance phagocytosis by macrophages. So we have two main opsonins. Can anybody tell me? One is
I
C3b and the other one is
IgG.
Excellent, doctor. Excellent. So we have two different types of main opsonins, IgG and C3b. So these will coat the bacteria and cause its phagocytosis. The next one is C3a, C4a, and C5a. These three. So remember these A along with C3, C4, and C5, and they will play a role in anaphylaxis. So remember that it will act in a anaphylaxis, and what happened in anaphylaxis, that how we will recognize that it is usually caused by these three, and it is the anaphylaxis. Usually the symptoms will be vasodilation and increased vascular permeability and histamine release. And among these three, C5a will be the strongest associated with anaphylaxis. Okay, doctors? And this C5a will also act as one more thing, that it will lead to neutrophil chemos chemotaxis as well. So it will, this C5a will attract neutrophil the last, the most important. So we have three different types option. Can you hear me, doctors?
Yes, doctor.
And my screen are visible now.
Yes.
So we have, so actually there are three main functions of complement system. Is first function is usually generalized, that it will, complement system will usually lead to inflammation. The second, the most important and the specific function, is opsonization. The main opsonins are C3b and IgG. And the third, the most important, is formation of MAC, and this MAC will usually lead to formation of pores inside the foreign particles, and it will lead to lysis of the particles. So the main component that are usually associated with MAC are C5 to C9. So these are the complement protein that are associated with MAC. So the MAC means cytolysis. So it will lead to formation of pores in bacterial membranes and will lead to cause cell lysis. And it is especially, this MAC is especially important for Neisseria species. So if person will have a deficiency of this MEC, then the person or will have increased susceptibility to Neisseria infections. Okay, doctors. and the next one is opsonins. So the two main opsonins are C3b and IgG, and its function is to enhance phagocytosis. We have some of the. Everybody clear so far? Can anyone have a
Yes.
question? So we have some of the complement inhibitors as well that will inhibit the complement system from activation and prevent the cascade of the complement system. So these are two different types of complement inhibiting protein. First is DAF, which is also called CD55, and the other one is C1 esterase inhibitor. So we know that C1 inhibitor are usually associated with classical pathway. So this will C1 is usually associated with classical pathway, and it will prevent the complement activation on self cells. And this DAF will protect host cell, especially, for example, RBCs. And if we a person will have a deficiency of this DAF, then what will usually happens? Can anybody tell me?
This is like that nocturnal.
Excellent paroxysmal nocturnal
Small,
No.
natural.
nocturnal hemoglobinuria. Okay, doctor. So this it is usually occur because of this DAF. We're gonna discuss in detail about paroxysmal nocturnal
Doctor, can you
hemoglobinuria.
repeat what the deaf do? What's this first
Actually,
sentence? The
so
first
actually,
sentence.
yes. So actually, the DAF is usually a complement inhibitor. So if this is usually in the patients, usually this DAF is usually deficient. So in this patient, there is increased risk of that they will develop paroxysmal nocturnal hemoglobinuria at night time, because at night time they are little bit acidotic, and due to this acidosis and they have a deficiency of this DAF. So in acidosis, the complement system are usually increased, and this complement system will then cause lysis of RBCs, and when they will wake up in the morning, their urine will turn black. This is called paroxysmal nocturnal hemoglobinuria, which is usually occurred in this patient because they will have a deficiency of a specific genes called pig A gene in these, which will act on this DAF. We're gonna discuss this in detail in a little while in the disorder of the complement. Okay, doctors.
Okay.
So prevent accidental destructions as well. So these are the two different types of complement inhibitors. So let's discuss the complement protein deficiencies first, and then regulatory proteins. So the complement protein deficiencies. So protein deficiencies is early complement deficiency. So these are C1 to C4. So this patient will have increased risk of severe and recurrent pyogenic sinus and respiratory tract infections, because this patient will have poor poor opsonization, because they will have a deficiency of C1 to C4, and we know that the opsonins are C3b. So we have also deficiency of C3b. So this patient will have poor opsonization and poor immune clearance as well. As well. And the patient which have a deficiency of C1 to C4 will have increased risk of development of SLE. Okay, doctors? Doctors?
Hello, doctor.
Yes, doctor.
So
Yeah.
this is
Hello, doctor. Please, what do you mean by poor immune clearance?
poor immune clearance, because opsonization is not only attached to these foreign antigen or foreign particles, they are also being attached to the dead and debris particles that are usually produced by the body own cells. So they will also opsonize these one as well, and then these are taken by the macrophages and will clear the immune system. So it will also act as a clearing particles as well. That's why they have two different types. Like first is opsonization of the foreign antigens and opsonization of the dead debris particles as well. That's why it is caused immune clearance. Okay, doctor?
Okay. Thank you.
Welcome. So early, remember this is the main MCQs and high yield point, that C1 2 and early complement deficiency will usually increase risk of SLE development in these individuals. And this is the high yield regarding exam point of views. So the next one is terminal complement deficiency. So it is from C5 to C9, and it is usually involved in activation of
Mac.
excellent MAC. And MAC is usually responsible for killing bacteria by forming
Or C.
excellent. So increase risk of Neisseria species or infections, and probably recurrent one. So the terminal are usually associated with recurrent infection with Neisseria. So these are complement protein deficiencies. Now complement regulatory deficiencies and regulation are usually done by
C1 inhibitor, and that's
excellent. So regulatory protein deficiencies. So the first one is C1 esterase inhibitor deficiency, and this deficiency will lead to
angioedema.
angioedema, because there will be uncontrolled kallikrein activation that will lead to increased production of kinin. Because bradykinins is then associated with formation of different clinical features like recurrent recurrent facial swelling and airway edema, because it will also lead to bronchospasm as well. And remember that these facial swelling and airway angioedema and these type of features, it is not associated with any type of urticaria. So no urticaria are usually occurred in C1 and no itching. So this is actually the differentiating features from other type of allergies as well. So the treatment is C1 inhibitor and bradykinin antagonist, and actually ACE inhibitor are contraindicated in these individuals. Okay, doctors, because ACE inhibitor will have increased risk of production of these bradykinins. So these patient will have not using these ACE inhibitors. So this is C1 esterase inhibitor deficiency. The second one is PNH, paroxysmal nocturnal hemoglobinuria. So actually, the cause of this is usually a deficiency of an gene called gene, PIG-A gene defect that will cause decrease GPI anchor. So actually there are two different types of proteins called DAF, which is called CD55 and CD59. So these are usually anchors by this GLP-1 and it is occurred through this gene. So when this gene is usually deficient, they will not anchor these two types of protein on the RBC's membrane. So the RBCs will become unprotected against complement system. So it is usually only been occurred when the complement system are usually activated, and I have told you that it is called paroxysmal nocturnal hemoglobinuria. So paroxysmal means episodic, nocturnal mean night time, and hemoglobinuria, hemoglobin in urine. So actually in night time when a person is usually sleeping, sorry doctor, so in night time when a person is usually sleeping, so he is a little bit slow breathing. So this slow breathing will increase the carbon dioxide retention in the lungs. So this retention will lead to a little acidotic the patient, and this little acidotic will lead to activation of complement system. And this complement system, as we know that these patients will have the deficiency of these genes, they will not be present with these DAF and CD59. So the complement system will then directly attack on these RBCs and will cause lysis of them. So the patient when wake up in the morning, he will have a black urine. This is called hemoglobinuria, and remember that it is not occurred in all the patient. It is usually occurred on those patient which has usually a deficiency of this pig A gene. Okay, doctors?
Okay.
Clear
Yes.
now, doc? Clear,
Yes.
everybody? So the complication of this paroxysmal nocturnal hemoglobinuria, and it is the most common cause of death in this patient, is Budd-Chiari syndrome
Hepatic thrombosis.
due to venous thrombosis. Okay, doctors. So this is the whole picture behind paroxysmal nocturnal hemoglobinuria.
Is
Okay,
there any
doctors.
treatment for this?
Yes, there
Is
is.
there any treatment for this
So
to prevent this?
so actually, we can also give different types of treatment, like to actually the main problem is usually to prevent this thrombosis, and this thrombosis can be prevented by giving the patient with a continuous dose of heparin in these patients, because the main life threatening event is actually thrombosis, not hemoglobinuria. Okay, doctors. So we should only been prevented these patient from hemoglobinuria. Okay.
So we can prevent the consequences, like we cannot fix the problem. That's the
Yes,
kind
yes,
of
because
genetic.
it is genetic problem. It cannot be
Oh,
fixed.
okay.
Okay.
Okay. Got it. Thank you.
Welcome. So it is most probably the risk factor that is associated that we should prevent these thrombosis. Okay. And we're gonna also discuss on detail this progressive nocturnal hemoglobinuria regarding its treatment options and investigations and also clinical features. Okay. Okay,
Okay.
doctor. So it is not only presented this preexamined hemoglobinuria with morning urine, it will also present with intravascular hemolysis, which will lead to decreased hepatoglobin, because that hepatoglobin will usually a binding site for free hemoglobin. So as a result of intravascular hemolysis, a free hemoglobin will occupy these hepatoglobins, and as a result, the level of hepatoglobins will usually decrease with us. So it is also a diagnostic one, and increase LDH with us and indirect bilirubin as well with us. Okay.
Okay.
Okay, doctors. So these are the other features. So you should only, because we are discussing here immunology, so in this you should only remember these DAFs, because these DAFs are usually responsible for prevention of RBCs from complement activations, because they are usually normally these cells are inhibit the complement systems. Okay, everybody clear so far, doctors?
Yes, doctor.
So
Yes.
let's move to the next one. So let's discuss the interleukins. So interleukins are cytokines. We're gonna tell them are cytokines that are produced by different types of cell, and their main action or function is usually mainly on leukocytes, means immune cells, and will cause activation of them. So the main source of interleukin 1 is, so the source for interleukin 1 is macrophages. Second source is monocytes. The third one is dendritic cells. So these are the main source for interleukin 1, and its function is to cause, this interleukin 1 is the main important one, is it will cause fever because it will act on hypothalamus and increase in hypothalamus a specific type of prostaglandins called prostaglandin E2. The second function of this interleukin 1 is increase adhesion on molecule on endothelium. Means it will also increase the adhesions of molecules on the endothelium, and it will also activate T cells, T cells, and it will also stimulate acute phase reactant formation from liver. So these are the sources, these three, and these are the function that is usually caused by this interleukin 1. Okay, doctors? So we're gonna discuss each and every one in this manner. So the second one is interleukin 2. The interleukin 2, the source of interleukin 2 is actually coming from activated CD4 T cells, and it will function of this is growth and growth factor. So actually this interleukin 2 is a growth factor for T cells first, and it will also maintain the regulatory T cells. Regulatory T cells that will prevent the overstimulation of the T cells. So these are called regulatory T cells. And the third function of these are stimulate natural killer cells. So this is the most important function of these, that they will stimulate the T cell proliferation. So remember that. Okay. So the interleukin 3, their source is actually coming from T cells, and it will have the function of this is usually growth of bone marrow, stem cells, and also support hematopoiesis. So remember from interleukin 3, so interleukin 3 is actually a bone marrow booster. The fourth one is interleukin 4. So interleukin 4 are released from the source of interleukin 4 are T helper 2 cells and mast cell, and their function is activation of IgE and IgG and differentiation of T helper 2 cells, and also macrophages activation. So remember from interleukin 4, it is usually associated with allergic reactions, allergic antibody, IgA. So remember that interleukin 4 have been strongly associated with IgE, allergic type of reactions. So the interleukin 5, its main source, it is coming from T helper 2 cells, and its function is growth and activation of parasitic cell that will work against parasitic type of infection, and those cells are called, anybody?
Eosinophils.
Eosinophils. Excellent. So remember from interleukin 5 eosinophils. And the sixth one is interleukin 6. And interleukin 6 is usually coming from macrophages and T cells, and its function is to activate the acute phase proteins, fuse, and B cell differentiation. So its function is acute phase protein, most commonly CRP, and fever and B cell differentiation. So this is a function of interleukin 6, and we have interleukin 8. Interleukin 8 are, their source is actually coming from macrophages and endothelial cells, and it will function of this is usually is neutrophil, chemotaxis, and neutrophil activation. Remember from interleukin 8, it will have a neutrophil migration. Okay, doctor?
Okay.
So, other one is interleukin 10, and interleukin 10 is usually released from regulatory T cells and macrophages, and its function is to stop the inflammation, and it acts as an anti-inflammatory, and it will decrease the T helper 1 cytokines, and it will also decrease the MHC class 2 expressions on the cell. So remember that interleukin 10 is a immunosuppressor. Then we have interleukin 13. The interleukin 13 is actually released from T helper 2 cells, and its the function is increase mucus secretions, and it will also airway hypersensitivity. So remember that interleukin 13 will cause asthma like symptoms. Okay, doctors. So these are the 13 different types of interleukins that are usually present, and they have different function with us. So remember that interleukin test 10 will have a immunosuppressor, and interleukin 3 will have asthma like symptoms, and interleukin 8 will have a neutrophil migrations because it will lead to chemotaxis and this, and interleukin 6 is usually associated with aqueous phase reaction, that is CRP. This is the main part in it. And interleukin 5 is usually associated with eosinophils, mean parasitic type of infections, and interleukin 4 is usually associated with allergic type of reactions because it will lead to activation of this IgE, and interleukin 3 will have a bone marrow booster, and interleukin 2 as a effect has a growth factor for T cell proliferation. So remember that. And interleukin 1 will have a fever and this type important effect. Okay, doctors?
What
Doctors,
was the name? Yeah, what
Yes.
the three was doing?
which one?
Three, three.
3 is actually the bone marrow booster. It will increase the bone marrow stem cells. Okay, so it is a
Okay.
booster. It will increase the stem cells like megakaryocytes and along with erythrocytes, and these different types of cells, monocytes and granulocytes. It will
Okay.
act
And
as a,
what was the difference in 4 and 5? They look similar?
the 4 is actually a IgE type, like allergic type of reactions, and interleukin 5 is eosinophil activators, so any type of parasitic type of infections. Okay, doctor,
Okay.
clear now?
Yeah.
So let's move to the important topic that is called respiratory burst. Let's explain this. Respiratory burst. So this respiratory burst is usually a mechanism is a rapid increase in oxygen consumption by phagocytes like neutrophils and macrophages after phagocytosis of different pathogens to generate reactive oxygen species that kill the microbes. So this is usually occurred in a phagolysosome, not in mitochondria. So remember that it is occurred in a lysosomal process. So the cell usually involved in respiratory burst are usually three different types. First is neutrophil, and these are most important one that will, and second one is macrophages, and the third one is monocytes. The purpose of them is to kill engulf bacteria and fungi using toxic oxygen radicals. So the stepwise mechanism of this respiratory burst is: first, bacteria is engulfed After engulfing, it will convert it into a body called phagosome, and this phagosome, when fused with lysosome, this is called, can anybody tell me?
Lysosome.
Excellent, lysosome. After that, an enzyme usually come. This is called step 1. And coming to the step 2. In step 2, an enzyme usually come called
PTH. Not pH.
NADPH oxidase. That is a key enzyme, and it is located in a phagolysosome membrane. So how it will usually react? It will combine with oxygen, and as a result, oxygen will have super oxide, and this is NADP plus. So this is the second step, and this step is usually the core of respiratory burst. The third one is then formation of reactive oxygen species from this superoxide. The step 3 is actually this superoxide conversion into different type of components with the help of an enzyme called superoxide dismutase. So this is converted into H2O2. This is the third step. So here 2 oxygen along with 2 hydrogen that will then convert it into H2O2, which is and O2. And oxygen are usually formed, and this is the third process, and the enzyme that are usually involved here is
This.
dismutase. So remember doctors, this enzyme is also important because the MCQs will also come on these enzyme as well, like conversion of superoxide into hydrogen peroxide require which enzyme? So you should go toward superoxide dismutase. Okay, doctors? So the step 4 is actually the most potent acid formation, which is called hypochlorous acid formation. And the enzyme required for this are myeloperoxidase, and it will require chloride. So how? That H2O2 will then combine with chloride, and it will then form hypochlorous acid and H2O, and the enzyme here is myeloperoxidase, and this hypochlorous acid will act as a bleach like compound, and it will kill bacteria by protein degenerations. Sorry, denaturation. And the last step is hydroxy radical formations. So this hydrogen peroxide is either converted into hypochlorous acid or either converted into hydroxy radical, which is also very toxic. So this is either converted into this hypochlorous acid, and it will also convert it into this hydroxyl radical, and this hydroxyl radical is also very toxic, and in this way the respiratory burst will actually kill different types of bacteria. So reactive oxygen species will destroy bacterial membrane, also DNA, also protein, and this will then lead to microbial death. Okay, doctor?
Okay.
So the oxygen dependent killing are usually oxygen dependent versus oxygen independent killing. So the oxygen dependent killing are respiratory bursts that will uses reactive oxygen species and are most prominent, most powerful, and it is usually occurred in neutrophil, most commonly macrophages and monocyte. And there are another types of oxygen independent killing, and those are lysozymes and granulozymes, which is usually occurred by cytotoxic T cells. Okay, doctor? So this is main point regarding respiratory burst. Everybody clear so far?
Yes.
Please, can you repeat the last statement, sir?
Which one, doctor?
The one you just said, I didn't hear you.
Adhesion.
Yeah, the last word, I didn't hear.
What was the other way of killing? One was the oxygen dependent. What was the other way?
The other way is usually actually there are two different types of killing of a bacteria. Is first is like oxygen dependent killing and second is oxygen independent killing. So oxygen dependent killing is actually occurred as a respiratory burst which will usually produce reactive oxygen species. So oxygen dependent, which
We
is
got this one, the oxygen one, but you mentioned another one without oxygen.
so without oxygen is actually a lysozyme and granulozyme. So these are actually the enzyme that are usually produced by CD8 cytotoxic T cell that will make pores in the cells, and as a result the water will move inside the cell and will cause lysis of the cell. This is called oxygen independent killing. Okay, doctors? So this is the oxygen independent killing. Now you are clear, doctor?
Yes. I got it, but I don't know about the other doctor. Maybe I
Thank
don't know
you.
what was her question.
Yeah, thank you.
Welcome. So this is the two types of oxygen dependent and oxygen independent killing, and this is actually not that powerful. Okay, doctors? Oxygen dependent killing is usually most powerful because it will form different types of acids, and this will completely eliminate the bacteria with us. Okay, doctors? So let's move to the important topic, that is hypersensitivity reactions. So can anybody tell me types of how many types of hypersensitivity reaction are present with us? Anybody?
Four types.
Four types. So,
Four.
can anybody define hypersensitivity?
Is body
Any?
immune reaction against any antigen?
So, uh, it can be taken as a hypersensitivity, but the correct definition is exaggerated or inappropriate immune response. Okay, doctor, exaggerated word is usually important.
Okay.
Exaggerated, exaggerated or inappropriate or abnormal response. So actually these inappropriate or abnormal response, but remember this exaggerated will be present in these inappropriate or abnormal that will cause tissue damage. This is called hypersensitivity, and we have four main types of hypersensitivity reactions. The first one is called type 1, and it is immediate type of allergic reaction, and it is IgE mediated. Okay, doctors. So the onset of this reaction is onset. Onset is immediate, like seconds to minutes. Remember that the time frame will also tell you about different types of allergic reaction, because the time frame is important here as well. Okay. So the mechanism of this hypersensitivity is that first exposure to it will lead to sensitization, means allergens like pollen, food, and drug. The most common type of food allergy is, can anybody tell me?
Nuts. Nuts.
Nuts, peanuts, वगैरह. Peanuts and other types of nuts. These are usually some people are very allergic to them. Actually, these will activate these T helper 2 cells, and these T helper 2 cells will then release three different types of cytokines. Can anybody tell me?
IL-5.
IL-5 and
IL-4 and 13.
excellent, doctor, excellent. So these IL-4 and IL-3 will produce IgE, and this IL-5 will stimulate eosinophil. Okay, doctors.
Okay.
So this IgE is then actually this allergen. This is the So this allergen like pollen, food, particle or drug will activate this T helper 2 cell. The T helper 2 cell will produce these three cytokines. In these three cytokines, these two will actually associated with production of this IgE, and this IgE will then activated or bind with Fc portion, actually Fc receptor. Remember that's Fc receptor on mast cell and basophils. And so this is the sensitization process. So whenever these different like pollen, food or drug are usually re-exposed to this, are exposure to this like allergen directly cross link with IgE, and this IgE will then lead to mast cell degranulation. And this mast cell degranulation will then release different type of like leukotrienes, prostaglandins, and histamines. And these three will then cause clinical symptoms like vasodilation, vasodilation, bronchoconstrictions, vasodilation, bronchoconstriction, increase vascular permeability, and the fourth one is also produce mucus production. These are the clinical symptoms that are usually present in this type of type 1 hypersensitivity reactions. And the example of type 1 hypersensitivity reaction, and it is important, the examples are, can anybody tell me the example?
Anaphylactic reactions.
Excellent. Anaphylactic. The second one is allergic
Asthma.
rhinitis that are usually occur due to pollens, and asthma that is and urticaria and food allergies. Okay, doctors? So remember from the mnemonic that ACAA, that anaphylaxis, C for cough, that means asthma, A for allergic rhinitis, and A for angioedema. Okay, doctors, that will lead to urticaria and this type of symptoms. So this is called type 1 type of hypersensitivity reaction. The type 2,
So, doctor, in the type 1, like when the first time the antigen comes, so they active these interleukin are releases and they bind with the antibody, and after the antibody activate this mast and basophil and these histamine releases. So this is for first event, but for the second event there will be no interleukin releasing, so they directly activate the antibody. Are you telling like that?
let me tell you, and then if there is any problem, so at the first exposure, that at the first time a person is usually exposed to pollen. So this first time exposure will lead to activation of these T helper 2 cells. T helper 2 cells here will produce different types of interleukins, and we have discussed that this interleukin will produce different types of function. Among these, interleukin 4 and 13 will produce IgE. This IgE will bind to the Fc receptor on mast cell and basophil. These are usually bind on these mast cell. So in the second, this first step is called sensitization. So on re-exposure, this IgE is already exposed, already attached to this mast cell. So whenever a allergen is usually again re-exposed this IgE, it will cross react with this IgE that is already attached to these mast cells, and they will directly lead to degranulations. Okay, doctors, because at this first step there is no more exposure to this IgE. That's why it will only attach and does not cause degranulations. And due to this allergic directly reaction with this IgE, that's why this process is usually in immediate. It will take seconds to minutes. That's why it is called an immediate process, and it is called the second sun. So this sensitization is usually the initial event, and after every time, like the first exposure and second, after first exposure, the all other exposure are usually re-exposure, and these are usually lead to this type of reactions. Okay, doctor?
Okay, so this sensitization is the first event. They don't need again and again. Okay, got it.
Doctor,
Okay, yeah, yeah, got it.
clear now?
Yeah, yeah. Thank you.
Welcome. So these are the two different types, that how type 1 hypersensitivity reactions, and these example are also being important, that you should remember this as well. The type 2 is usually take, it is usually an antibody mediated, and it is called cytotoxic, and it will usually take hours to days. It will, its onset is hours to days, and its mechanism is these IgG and IgM antibodies. These IgM and IgG antibodies will attach on the cell surface. Antigen, cell surface antigens, and also on the basement membrane and on receptors. We're gonna also discuss the different types of diseases that on like receptors. Can anybody tell me an example of this type of hypersensitivity reaction that these antibodies will attack the receptors?
Like
It is a
Misthania
very common
gravis?
disease,
Like Misthania?
and we have discussed 2 days earlier the test regarding this disease.
Misthania gravis is also an example.
Like IgA nephropathy. What happened?
Yeah, I think maybe his connection lost. Can you
Explain.
hear me, Iman?
Yeah, yeah.
Okay.
You say the myasthenia gravis, but we couldn't hear him at that time.
Sorry, doctor, sorry. It's actually a problem that is continuously coming. So where were we, doctors?
We're
MG.
talking about type 2 hypersensitivity reaction.
So the type 2 hypersensitivity reactions are usually antibody mediated, and their onset is usually from hours to days, and its mechanism is actually these IgG and IgM antibodies that will attach on the cell surface antigen and basement membrane and receptors. And can anybody tell me that these receptors are usually affected in which disease?
Good pastor.
Good pasture and which other disease?
Autoimmune hemolytic anemia.
Sepsis.
Myasthenia gravis.
Myasthenia gravis and good pasture and these type of disease. We're gonna also discuss the example as well. So this is the mechanism that these antibodies IgG and IgM will attach on the cell surface antigen basement membrane and in receptors. So the basement membrane will actually lead to good pasture syndrome and these. So these target of these are, so as a result, so this is the mechanism and as a result it will lead to complement activation through which pathway?
Classical.
Classical pathway, excellent. And opsonization because IgE is a main
Obstinance.
opsonance and it will also lead to a cellular cytotoxicity. So these
Doctor,
are,
for
yes.
me, I don't know if it's for everyone, but this AD, we didn't hear this after opsonization.
So ADCC is actually antibody dependent cellular cytotoxicity. So the antibodies that will lead to cellular distractions that will require these antibodies. And the fourth one is receptor dysregulation or dysfunction actually. So we're gonna, so this will lead to these three, four types of symptoms and their targets are main cells or tissues. The example of cytotoxic distractions, cytotoxic destructions, that is cell destructions. These are, now you guys can tell me the examples. What are the cytotoxic destructions that is usually caused by these antibodies? Autoimmune hemolytic anemia first. Second one is
Transfusion
good
reactions.
transfusion reaction, excellent, and goose pasture syndrome. Third one is rheumatic fever. And the fourth one is hemolytic transfusion reactions. And the receptor dysregulations or dysfunctions are the following. First one is Graves' disease. Grave disease. And this grave disease is actually occurred due to receptor hyperstimulation. Okay, doctors. And in myasthenia gravis, it will lead to receptor blockage. because it will dis. And the other one is pernicious anemia. So it is also come under second type 2 hypersensitivity reactions. So we have these type 2 hypersensitivity reactions. It is mainly caused by antibody mediated, and these antibodies will then lead to destruction of different type of and this destruction is usually occurred via complement system and opsonizations and antibody dependent cellular cytotoxicity and receptor dysfunction. And their example are autoimmune hemolytic anemia, Goodpasture syndrome, rheumatic fever, autohemolytic transfusion reactions, and grave disease, myasthenia gravis, and pernicious anemia. Okay, doctors. So the next one is type 3. Type 3 is actually occurred as immune complex deposition. Okay, doctors. So the immune complex depositions, and it will the onset of this is 1 to 3 weeks, and 1 to 3 weeks, and its mechanism is that at antigen antibodies complexes forms, and they will circulate in blood and then deposited in tissues like blood vessels, kidneys, joints, and skin. And then after deposition, it will activate complement system and will cause this complement system will then cause activation of inflammation and also tissue damage as well. So this is the mechanism of type 3, and the tissue damage is usually occurred by these neutrophils. These neutrophils are usually lead to tissue damage, and the example of type 3 hypersensitivity reactions are, this is important, SLE, post streptococcal glomerulonephritis. Third one is serum sickness, and fourth one is arthros reaction, and fifth one is polyarthritis nodosa, which is a medium vessel vasculitis. So this is type 3 hypersensitivity reaction. The fourth one is type 4, and the type 4 is usually,
Doctor,
yes.
can you take the examples that you gave again? I didn't quite get it. The last three for the
Which
type
one,
3.
doctor? The
typically
last,
the examples, yeah, the last two that I gave the examples.
here, this is called Arthur's reaction.
Trust
And
reaction.
this, yes, and this fifth one is polyarthritis nodosa, which is a medium vessel vasculitis. Okay, doctors, that will spare lungs and involve GIT, nervous system, CNS, and liver, and it will be associated with hepatitis B. So this will involve these three. So the patient presented with pain will have melena and these type of symptoms, and it is usually occur due to accumulation of these immune complexes into the vessel wall, and as a result a vasculitis is actually take place then. Okay.
Which system is not involving in the pen?
Pan, remember, in pan no lung involvement are usually occurred, lungs are usually spared. Okay.
Okay.
So in pan lungs are usually spared, remember that. Okay, doctor,
Okay.
clear now?
Yes.
So the type 4 is the mechanism of type 4 is that the onset, first discuss the onset. So the onset are usually 48 hours to 72 hours, and its mechanism is no antibody involved and mediated by CD4 T helper 1 cell and CD8 cytotoxic T cell. Actually, these cells will produce a cytokine called interferon gamma, and this interferon gamma will activate macrophages, and these macrophages will then lead to tissue damage. So this is type 4, and the type 4 are usually a delayed response and and the example of type 4 hypersensitivity reaction is important is PPD test, which is tuberculin test, and contact dermatitis, which is usually due to nickel, silver, and poison ivy, and type 1 diabetes mellitus, multiple sclerosis, and graft versus host rejection. So remember from that is that T cell will take time. That's why it is called type 4, and it is a delayed type of hypersensitivity reaction. So the first one is usually immediate, the type 2 is usually will take hours to days, and type 3 will usually take days to weeks, while type 4 will usually take around 48 to 72 hours. Okay,
Okay.
everybody clear?
Yes.
So remember from these four T's as well. Like first T is it is actually involved T cells. It is involved in transplant rejection. It is also involved in TB, skin contact as touch or touching means contact dermatitis. So these four T's, remember from this, it is associated with type 4. Okay, doctor? Everybody clear?
Yeah, what is this DRESS, the drug reaction with eosinophilia and systemic symptoms? So, is there any mnemonic? DRESS.
Yes, address means so address means drug reaction with sorry drug reaction with eosinophil and systemic symptoms. This is usually DRAS. Okay, doctor? So in DRAS, the patient are usually will have presented with skin rash, eosinophilia, and internal organ involvement. Okay, and it is also type 4 as well. Hypersensitivity as well because it is mediated by these T cells. Okay,
So, is there is no specific organ, so it can involve any organ.
any
Like, so,
organ, an
okay.
internal organ involvement, any organ can be involved in it, and it can, as its name implies, here eosinophil will be raise, and there are systemic symptoms are usually being present with that, like systemic symptoms. Can anybody tell me?
Like renal involvement or liver involvement.
Yes, like renal involvement, kidney involvement, lung involvement, heart involvement. So, like liver will have hepatitis, and kidney will have interstitial nephritis, lung will have pneumonitis, and heart will have myocarditis, and CNS involvement can be also occurred in stress as well.
Okay.
Okay, doctor?
Yes.
Okay.
Clear now?
Yes, thank you.
Welcome. So this is the last topic of our today's session. Tomorrow we are going to discuss some of the transfusion related reactions, and after that we will discuss the uh, transplant rejections, and after that we have to discuss some autoimmune diseases and also immunosuppression. So tomorrow we will cover these four topics, and then we're gonna end our immunology, and a day after tomorrow then we have to cover our MCQs. Okay, it will also take 2 or 3 days.
Inshallah.
Okay, doctors,
Okay.
everybody
Okay.
clear?
And what about the vaccines? We are not going to do the vaccines?
The vaccine topic is not done, so we will also done the vaccines tomorrow. Okay,
Okay.
sorry, I have forgot the vaccines. So the tomorrow we gonna cover these 5 topics vaccine along with all the 4 topics as well. Okay, don't worry.
Thank you.
Welcome. So this is the last topic of our today's session. So see you tomorrow, guys. Bye bye.
you. Thank you, doctor.